So in the second part, we will continue our compartmentalization of the cell, and on an average, the membrane enclosure compartments together occupy basically half the volume of a cell, as I told you earlier, and a large amount of intracellular membrane is required to make them all. For example, in liver and pancreatic cells, the endoplasmic reticulum has a total membrane surface area that is almost 25 times and 12 times respectively that of the plasma membrane in the same cell. So in terms of the area and the mass, the plasma membrane is only a minor membrane in most of the eukaryotic cells. So that means the other membrane enclosed organelles or compartments make up a significant share of the eukaryotic cell. So this was another aspect of the cell compartmentalization. Similarly, the membrane enclosed organelles have what is called as characteristic position in the cytosol. It is not something is random. They have a position which is unique to every structure in most cells. For example, the Golgi apparatus is located close to the nucleus, whereas the endoplasmic reticulum will extend from the nucleus throughout the entire cytosol. So these characteristic distribution patterns depend upon the interaction of the various compartments or the organelles with the cell cytoskeleton, because you know cell cytoskeleton is very important to render the what is called as basic structure of the cell intact. So the localization of both AR and Golgi apparatus, for instance, will depend on their intact what is called as microtubular structural framework, because it has been seen that if we use certain drugs which can basically depolymerize certain microtubules, what happens? The Golgi apparatus, the fragments, they disperse throughout the cell, and the endoplasmic reticulum may even collapse. So that shows us that the cell compartmentalization is sustained by the cell cytoskeleton, mostly composed of microtubular framework. Microtubular framework. Likewise, when we talk of the this compartmentalization of the cell, or the what is called as compartmentalization of various organelles, what we basically know from the origin or evolutionary point of view, where from these organelles or compartments have evolved. So that the evolutionary scheme basically tells us that the intracellular compartments in eukaryotic cells, they basically fall into four distinct families as per their evolutionary history. For example, number one, the nucleus and the cytosol, which communicate through nuclear pore complexes, are topologically continuous, although they are functionally distinct. They make a single group as per their origin, so they will say they are topologically continuous. Second is the all organelles that function in the secretory and the endocytic pathways. For example, endoplasmic reticulum, Golgi apparatus, endosomes, lysosomes, and other class types of numerous intermediates like transport vesicles or peroxisomes. They make up a separate group of organelles or compartments which is thought to be a single group. Similarly, the mitochondria is the third one, and the plastids are separately considered because the origin of the mitochondria and plastids is considered to be of endosymbiotic nature. It is being considered that they were first bacteria-like structures, which basically then resulted in the formation of these typical mitochondrial and chloroplast organelles in eukaryotic cells. So because of this differential occurrence of various groups of organelles or compartments, it basically helps us to understand how the proteins then move between different compartments, and they move into different ways. As I told you, that these things we have to study later on in the cell biology, so we will not be touching them today, how the protein sorting takes place, or how the cell signaling operates, how are the different fundamental ways when the proteins move from one compartment to another compartment. So these mechanisms we will take up later on, but in this audio we need to stress on the what is the relationship between the compartments of the cell, and how to consider that the different organelles or compartments may have evolved. As you know, the precursors of the first eukaryotic cells are thought to be the simple organisms like bacteria. which generally have a plasma membrane, but no internal membranes or no internal what is called as compartmentalization. So the plasma membrane in such primitive cells therefore provides all membrane-dependent functions, including proton pump, ATP synthesis, protein secretion, lipid synthesis, so on. But as we see this gradation in the eukaryotic cells, we don't find that all these functions are performed by the plasma membrane itself. No, in the present day eukaryotic cells, which are 10 to 30 times much larger in dimension and 1000 to 10,000 times greater in volume than a typical bacterial cell like E. coli. So the profusion of internal membrane system can be seen as an adaptation to increase in the cell size. Therefore, the eukaryotic cells have a much smaller ratio of surface area to volume, and its area of plasma membrane is presumably too small to sustain the many vital functions for which membranes are required. So that basically tells us how the different compartmentalization events or different organelles might have evolved in the eukaryotic cells to compensate those deficit functions which are basically performed by the plasma membrane only. And therefore, the evolution of internal membranes evidently is accompanied by specialization of different membrane and compartment functions. For example, the generation of thylakoid vesicles and chloroplast, although this is not the part of the animal cell, but you see how the thylakoids are created in the chloroplasts and what are the different functions of these thylakoids in case of plants, right? Or if you talk of the mitochondria matrix, the mitochondria, what is called as the grana, the mitochondria, the inner cristae, how they are specialized to perform different functions like the thylakoids or the chloroplasts. So that means the compartments in eukaryotic cells have originated in a conceptually similar way, basically. However, they pinch off specialized intracellular membrane structures from the plasma membrane, right? So that means we say the origin of these or compartments or organelles or this topological relationship must be that they might have originated from the membrane itself. We can therefore think of all these organelles as members of the same family, like the organ, whatever it is. So we may say like the Golgi apparatus, the ER, endosome, lysosome. So they might all have originated somehow from the plasma membrane like structure. So later on, we will sorry, you will study the much about this cell compartmentation from the text material I have uploaded, and but this voice recording will help you to further understand the concept of compartmentalization.